Okay, good morning. So we spent a lot of time in the past few lectures talking about how you can use linkage analysis or genetic association mapping to find a marker that is linked or segregates with a trait of interest. So that gets us into the ballpark of where the gene that you're interested in is. So today we're going to spend a fair amount of time talking about how you can actually identify the gene of interest after you do this, perform some sort of linkage analysis or genetic association study. And then what we're also going to talk about a little bit today is we're going to talk about how many of the techniques that involve linkage analysis are being supplanted by new sequencing technologies that are allowing us to basically just identify the disease-causing mutation directly from sequencing. So you can skip a lot of the earlier steps of things we talked about. And lastly, we'll end the lecture talking about once you've identified a disease-causing mutation, how you can maybe intervene on that disease-causing mutation, make some sort of gene alteration to correct the mistake. And one of the approaches we'll talk about that you can use to do that sort of thing is CRISPR-Cas9 genome editing, which you're probably hearing more and more about in the news. So what I'm showing you here on this slide, I'll talk more about this later because I don't really have time to do justice to it right on this slide. It would be too complicated. But basically, this is a result of doing a sequencing reaction using a new sequencing methodology. And basically, each of the spots represents a different clone of a DNA molecule that's being sequenced. And the process involves capturing images at sequential time points to sort of determine what the sequence is. Again, this doesn't make any sense at this point. It's just a pretty picture. We'll talk more about that on a later slide during this lecture. Okay, so you've gone around and done some sort of linkage analysis, either a pedigree-based linkage analysis where you're looking within family, or some sort of population-based genetic association study. And you've identified a marker, which in this figure is designated by this little green box. So some sort of molecular marker that you find is linked to the gene of interest. And the gene of interest, let's say, is some sort of disease gene, so it has some sort of mutation in it. And so basically what that does is it kind of gets you into the ballpark of where the gene is. So once you've identified this linked marker, what's next? What do you do next? Basically, how is it that you would use this information to identify the gene of interest? So it could be that this linked marker lies basically millions of base pairs away from the gene of interest. So you could still be a long ways away from the gene of interest. You're much closer than if the gene could be anywhere in the genome. You've gotten it mapped down to a fairly small interval, but you could still be millions of base pairs away. And the other thing is that there may be many other genes that reside in this region. So being able to figure out which of these genes is the gene you're trying to map the marker to could be quite a daunting task. So basically, the approach that's used to identify the gene of interest is that you actually sequence the entire region, and then you use a variety of approaches to figure out which of these genes in that region is actually the disease gene that you're searching for. Oh, and by the way, I've got the term disease gene in quotation marks because, in fact, the term disease gene refers to a gene that we probably all have. It's just that in some of us, if you have a mutation, it gives rise to a gene. Whereas if you don't have that mutation, it doesn't give rise to a disease to the disease. So it's a gene we probably all have. It's just that it causes a disease if it has a particular mutation in it. So again, there's a variety of approaches you can use to identify the disease gene. We'll talk about that with a specific example today. One way you could do that is by comparing the sequences of all these genes in cases and controls, cases being the people that have the disease, controls being people who don't, to see if you can find a sequence difference that reveals where the mutation resides and which gene that mutation happens to reside in. There are challenges with that as well. We'll talk about that more towards the end of the lecture. Okay, so the specific example I'm planning to talk about today is basically the identification of the BRCA1 gene. Mutations of this gene give rise to a heritable form of breast and cervical cancer. Okay, so the term that we're going to be using to describe this example is positional cloning of the BRCA1 gene. The reason this is called positional cloning is because basically it involves first mapping a linked marker to the trait of interest. And once you have that linked marker that's near to the trait of interest, then basically that gets you into the position of the gene. And then once you're in the position of the gene, you can use that information to eventually identify the gene through methods we'll talk about today. So again, this is called positional cloning. So the work that was done to identify the BRCA1 gene was done by Mary Claire King, who's actually a faculty member in our department. And the work that she started to identify this gene was she identified a number of individuals in the population who seemed to have a genetic form of breast cancer and cervical cancer. So she identified a number of pedigrees of people who seemed to be segregating this trait in a fairly simple Mendelian fashion. And so she just basically performed linkage analysis to identify linked markers that would segregate with the trait of interest. And then she used that information to identify the gene. So the steps that are involved in positional cloning go as follows. Basically, you want to identify families that segregate some sort of heritable form of disease. That's important. You need to know it's heritable. Then you perform linkage analysis with a large collection of polymorphic markers to identify the genomic position where the disease gene resides. And then ultimately, once you get into the ballpark, you can sequence the genomic position containing the disease gene to identify the disease-causing mutations. Now, as I'll talk about a little bit later, this work was all done before the genome sequence was identified. So Mary Claire King actually had to do some things to identify this gene that we really don't have to do anymore. So some of the things she had to do to identify the disease-causing mutation are things that I'll talk about very quickly because they're mostly historical, but I'm not going to go into great detail on them, and I don't expect you to regurgitate all this information on an exam, but I just want to sort of present some of this because some of it's historical. But some of the logic involved in this is actually relevant to even things that are done now. Okay. So how did linked molecular markers help identify the BRCA1 gene? So these are the name of two different markers that Mary Claire King found were linked to the trait of interest, D17S1321. I won't say what the other one is. I just want to make sure I was clear. This is the names of these linked markers. These are sites in the genome that are polymorphic in the population where the sequence varies from individual to individual. So basically, she identified two markers that seemed to flank the BRCA1 gene on chromosome 17. But the sequence, again, the sequence of the genome was not known, so the sequence that resided between there was completely unknown, and the location of the disease-causing mutation was also unknown, okay? So this sequence is all unknown. All she knew was that the BRCA1 gene was somewhere in that interval. She also knew that based on the linkage mapping results, that this interval consisted of 0.6 centimorgans. And if you recall something that I said in an earlier lecture, one centimorgan in humans is 1 million base pairs. So the fact that this interval is defined by these two markers was 0.6 centimorgans. That says that this sequence here must have been about 600,000 base pairs. So a lot of DNA sequence in here, probably containing multiple genes. So basically, the important point here, though, is that because they were using molecular markers to map the location of the BRCA1 gene, we actually know the sequences of the molecular markers because at that time, there were just portions of the genome that were sequenced and compared in different individuals to find polymorphic markers. So these sequences are basically landmarks. They just have a short bit of sequence here, but these are starting points to begin to use these sequences to identify the intervening sequences through methods I'll talk about in the next couple of slides. So, and the basic goal here is basically to retrieve the unknown sequences between these two markers. And the way you do that is basically you use these known sequences as radioactive probes to screen a genomic DNA library. Again, I'll just go through some of this fairly quickly because you've talked about how to prepare a genomic DNA library. That basic approach that you talked about in the first half of the course will be used to create the library that we're using in this study as well. And then you identify the gene within the clone sequences using a variety of different approaches. For example, by looking for mutations that distinguish the cases from the controls, basically. Okay, so let's talk about how to retrieve sequences surrounding a molecular marker. Okay, so again, the way this is done is that you actually make probes from these known sequences. These are the sequences that correspond to the polymorphic markers. And then what you do is you create a genomic DNA library using methods that you talked about in the first half of the course. I won't go through this in any detail, but these are basically figures from the first half of the course. But you use the same procedure to create the library. And then what you do is you basically use these probes, you make them radioactive, and you use them as hybridization probes to screen clones in the library to figure out which of the clones have sequences that are complementary to the probe sequences that you're using. And then once you identify some clones in the library, DNA clones from the library, you make those new DNA clones radioactive and you use them to screen the library again. And basically what you do is you progressively find overlapping inserts so you can actually identify larger and larger chunks of DNA in the region. and again, and then you just repeat this process over and over again. So I realize this, I'm going through this fairly quickly, and this slide doesn't make the issue very clear. The next slide should clarify things a little bit, but I'm not going to get into too much of the detail of this because, as I mentioned at the beginning, this is really sort of a method that is not used so much anymore. It was used heavily for many, many years, but it's being supplanted gradually by other approaches. So I just want to talk about this mostly for historical purposes. So this basically the slide describes how chromosome walking works. Let's say these are two markers that flank the gene that you're trying to identify. And again, because we're using molecular markers in this analysis, you actually have the sequences that flank each of these polymorphic sites. So what you do is you take the DNA sequences that correspond to the markers you're using in your gene mapping experiment, and you make them radioactive. That's supposed to be designated by the fact that they're red. And then you use them to screen a library. And the way you do that, I'm not going to describe this in any detail, but you're basically using hybridization to find clones in your library that basically have sequences in common with these markers. So you use these as hybridization probes to screen the library. And again, like I said, don't worry about this because I'm not describing this in any detail. I'm just trying to give you the overall picture of how this works. And let's say these are the first clones that you pull out of your library. So, and the clones are going to be bigger than the probes typically. It depends on the size of the clones you make in the library. But let's say these are the first clones you pull out of the library. And then what you do is you basically take these first clones you pull out of the library and you make them radioactive. And then you rescreen the library. And the goal here is you're searching for additional clones in the library that overlap with these sequences and that will further extend the sequence for you. So again, each time you pull out a clone from the library, you make it radioactive, and you basically use it to rescreen the library. So let's say on the left, you used that first clone from the library, you made it radioactive, you rescreen the library, you pulled out some additional clones from the library that overlap it. So you can see you're kind of extending the sequence. The goal is to extend the sequence towards the other marker, but you actually, typically when you do this, you walk in both directions. I'm just showing you this, that you're trying to walk towards one another. You don't really know which way you're going when you're actually doing this analysis. And then with the other library clone that you pulled out, you find some additional sequences that overlap with it. And you just keep going. You make each time you pull out a clone from the library, you rescreen the library the goal being to find additional clones that overlap with it. And you just keep going with this process until eventually you find clones that meet with one another. So each time you pull out a clone, you sequence it. And then eventually you're going to find that you're going to pull out clones where the sequences overlap. And by doing this, you're basically going to get all the sequences between the two markers that you use in your gene mapping experiment. So, and again, this is called chromosome walking because essentially you're just walking towards the two markers that flank the gene of interest. So using chromosome walking, Mary Claire King's lab actually retrieved all the sequences that lie between these two linked markers, the BRCA1 trait, and then they sequenced all this DNA. And basically, they end up with a sequence that looks sort of like this. This is just a small part of it, though, because to show the entire 600,000 base pairs that lie in this region, I'd have to have another 769 pages of this this length to really reveal all of that. So what do you do at this point? So basically, there's a bunch of different approaches you can use to identify genes in this region. So let's consider some of the approaches. Basically, one approach you could use is you could look for sequences that are indicative of promoters. Again, these are sites where transcription begins in the gene, and typically many promoters have conserved sequences in the promoter regions that are recognized by transcription factors that initiate transcription. So many promoters have sequences that are in common. You could also look for transcribed regions, and one way to do that is by making a cDNA library. I don't think you talked about this in the first half of the course, but cDNA libraries are basically just libraries where you collect all the messenger RNAs that are made in the cell, you convert them to DNA, and then you clone them into a plasmid, and then you can actually sequence those, and you can map these cDNAs onto this genomic DNA to figure out where all the open reading frames are, or the coding sequences are. Another thing you could do is just look at the sequences to search for open reading frames. If you see a large stretch of an open reading frame, just by chance, if it's a very long stretch, it's unlikely to just be there by pure chance. So if it's a really large open reading frame, it's likely to be a protein coding sequence. Okay, so let's say you performed all these analyses, you've identified all the genes in this region that are flanked by the two polymorphic markers that are segregating with the BRCA1 trait. You've mapped it to a region, you've identified all the candidate genes in the region, but we still don't know which of these genes is the gene of interest. So how can we tell which of these is the BRCA1 gene? what kinds of reality checks could we do to sort of say, oh, this makes sense. This is what we'd expect from the gene based on what we know about the trait. So here's some possible strategies. So for example, you could ask, is the wild type version transcribed in the appropriate tissues? Depending on where you see the trait phenotype, that's the tissue you might expect the gene to be transcribed in. So for example, this mutations in BRCA1 give rise to breast cancer, so you might expect this gene to be expressed in breast tissue. You could also ask, does the predicted function of the protein make sense? So there's lots of biological functions that cells carry out, but you might be able to predict what kind of biological function might be involved in cancer, for example. Since I haven't discussed cancer, I can't really be hard for me to sort of give you an example of what to expect there. But after the cancer lecture, I could probably give you some genes and say, does this be likely to be involved in cancer? And you'd be able to say yes or no. Does the gene recovered from patients have mutations relative to the version from normal controls? Because again, if this is a gene that's associated with disease, presumably that's the case because it bears some sort of mutation. So that's the sequence of this gene should look different from the gene in normal individuals who don't have the disease. Another thing you could do is you could ask whether introducing a mutant allele into a model system causes similar phenotypes. So if you identify a mutation you think is responsible for the disease, if you create that mutation in the corresponding gene from, say, mice, do you see mice developing cancers as well? So ultimately, the BRCA1 gene was identified, and it was actually first identified from Mark Skolnick's lab in 1996. And the evidence he presented to say that this was actually the BRCA1 gene is as follows. He found that the gene itself was expressed in breast and ovary tissues. So that was what you would kind of expect from a gene that causes breast cancer and cervical cancer. He also found that it encodes a really large protein, 1,863 amino acids in length. He also found sequence differences between normal subjects and families that segregate breast cancer. So for example, the kinds of things he detected in patients were things like an 11 base pair deletion beginning at codon 24. So that's an out of frame deletion. So after codon 24, you wouldn't make the rest of the protein. He found a glutamine to stop codon at codon 1313. He found a 1 base pair insertion at codon 1775. So that would be, again, a frame shift mutation. A T to G missense mutation at codon 2099. And in some families, he found that no transcript was made. So I should emphasize right here that these are different mutations that were detected in different families that were segregating this disease. So each of these mutations must have risen independently in these different families. So they had the same trait, but the trait was caused by different mutations in the same gene. And then he looked at the frequency of these various mutations he detected in the patients in controls. And what he found was basically that their frequency was basically just completely absent in control samples. So he never saw any of these mutations in any individuals that did not have breast cancer. Okay, so I'm going to come back to the BRCA1 gene when I talk about cancer in a later lecture. So we'll return to this gene later to talk about what it does. So the BRCA1 gene was identified in 1996, and it's now 2020, so a lot of things have changed over that span of time. So back in 1996, when the BRCA1 gene was first identified, you could basically identify in the order of hundreds of base pairs of DNA from a given instrument in a single day. So, and this was actually the method that was employed at that time is known as Sanger sequencing. That was the basic method. But if you come to the present, basically, and this is actually a little bit out of date, now you can sequence something on the order of 500 billion base pairs of DNA in a single day. It's pretty incredible how much the technology has changed. And this has really dramatically impacted how we can identify disease-causing genes. We'll talk about that a little bit later in the lecture. So I want to first talk about a little bit about the sequencing developments and talk a little bit about the Human Genome Project before I sort of go on and talk about the sequencing technology that's being used currently. So this is a chart of the cost per genome per year, and that's shown in the green line on this chart. And this is being compared to Moore's law. Moore's law is not actually a law. It's a sort of a benchmark standard that's used by the computer industry, which basically says that computer power should double every 2 years for the industry to be doing really well. So it's a benchmark that most industries have a hard time matching. So what you can see is starting around 2001 up to about 2007, 2008, the improvement in genome sequencing capacity was improving steadily, or at least on the cost basis. And it was following Moore's law, which is actually pretty darn good. But suddenly, right around 2007, 2008, it really takes off. So something changed dramatically at that point. So we'll talk about that a little bit later. So again, I want to remind you, this chart begins at 2001, but in fact, the BRCA1 gene was identified in 1996. So things were really not very efficient back in 1996. And then a little bit later in the lecture, we'll talk about what happened in 2007, 2008 that led to suddenly this rapid development of improvement in sequencing technology. But before I talk about either of these things, I want us to say a few words on the Human Genome Project. So the Human Genome Sequence was completed, mostly completed, in around 2001. There's a couple of papers that sort of published some of the initial findings from the Human Genome Project. And this is a question that works a lot better when I have a live audience in front of me, so I'm not going to belabor this point because this is not something you have to spend a lot of time thinking about. But this is more out of my own curiosity. I'm just curious what people sort of tend to think when they come into the class in terms of how many genes humans have. You can just take a guess at this, but I'll tell you the answer right straight away. It's 20,000. That's roughly about how many genes humans have. And then I have another question on the next slide. Again, these questions are not things you need to spend a lot of time thinking about. These are just for my own sort of curiosity and interest. It's kind of getting a sense of what people think. So which organism has the most genes? And if you think it's humans, 20,000 genes is what humans have. You'd be wrong about that. You might think, for example, it's C. elegans. If you guess that as the organism that has the most genes, you'd be wrong once again. What about this plant, Arabidopsis? That plant has about 25,500 genes, but if you pick that one as your top choice, you'd be wrong again. It turns out the species that has the most genes is this water flea, Daphnia, which has 31,000 genes. So this is probably a surprise for lots of people. Generally, we tend to think of ourselves as being at the top of the evolutionary tree. But this view of humans being at the top of the evolutionary tree is the wrong idea in terms of gene number, at least. We're just one branch on the evolutionary tree, and the number of genes that a given organism has just really kind of depends on the environment and their needs. It's not a direct measure of the complexity of the organism itself. But it turns out genes are actually only a small part of the human genome. So if we break down the human genome to try and figure out like how much of it codes for genes, it turns out for protein coding genes, they only account for about 60 million base pairs. Now that sounds like a huge number, but in terms of the overall size of the human genome, it represents only about 2% of the base pairs in the human genome. So the protein coding portions of the human genome are represented by this blue slice of this pie. So only 2%. Now another 3% consists of regulatory elements. These are like promoter sequences, for example, or sites and introns that are involved in splicing. But basically, regulatory sequences in the human genome account for another 3%. So that takes up about another 90 million base pairs. But that still leaves 95% of the genome completely unaccounted for. It's 95% of the human genome doesn't perform any known function that we're aware of, basically. So let's break down this 95% to try into different categories. So it turns out much of this non-coding sequence, non-regulatory sequence, consists of transposons. And transposons are just essentially some of the simplest parasites that exist. They're just short sequences of DNA that have the ability to insert themselves into the genome. And then they also have the ability to make copies of themselves and cut themselves out of the genome. So basically, they go through this repeating process where they make copies of themselves, they insert themselves in the genome somewhere, they make new copies of themselves, they insert themselves in the genome again, so they can repeat this process. So it turns out transposons represent about 45% of the non-coding sequence in the human genome. And there's two types of transposons that predominate. One type is these line elements. These are about 6 kb each. There's probably about 900,000 copies of these in the genome. These account for about 15% of the human genome. And then another 1.5 million copies of another type of transposon called sign elements. These are much smaller, about 100 to 300 base pairs, and they account for about 10% of the human genome. So it's kind of a weird feeling to think that about half your genome consists of essentially a parasite that just replicated itself repeatedly in the genome. What about the rest of this? So about 25% of the genome is introns. So protein coding sequences represent, again, about 2%. They're heavily dwarfed by the size of the introns. The introns take up much more space than the protein coding sequences do. And then there's about 15% that's just repetitive DNA, these repeats. Many of the repeats are at the centromere, but they're just repeats of the same DNA sequence, like CAG, CAG, CAGs, things like that. There's lots of different kinds of repeats, but about 15% of the genome overall is represented by these repeat sequences. And then the last 15% is just unique non-coding DNA. So this is the genome. Very small percentage of it's devoted to protein coding sequence. Okay, so now let's talk about what happened here. Why suddenly the cost of sequencing really sort of dropped precipitously. And basically what happened here was that there was the development of what's known as massively parallel sequencing. You can sequence many, many, many DNA molecules simultaneously using these really simple approaches that were developed. So there are several different types of massively parallel sequencing platforms that are being used by the research community. I'm listing two of the main ones that are used by the research community on this slide here. And by the way, massively parallel sequencing is sometimes called next generation sequencing, or simply NGS. So again, I'm only listing two of the ones that are used by the research community on here, but there's many others that are in development, and I'm only really going to talk about one in today's lecture, the very first one, Illumina's Luxus sequencing by synthesis. Okay, so let's talk a little bit about that. Okay, just a brief summary about what you can do with massively parallel DNA sequencing and some of its features. Some of this will make more sense when I go through and describe how this all works in the next few slides. But basically, massively parallel DNA sequencing allows you to generate hundreds of millions of sequence reads. And again, what a read is, I'll talk about that a little bit. It's about 10,000-fold cheaper than Sanger sequencing. And my slides are a little bit out of date, so it's probably actually quite a bit cheaper now than it was a couple of years ago when I put this together. But there's some limitations, or at least some challenges to have to deal with. The sequence reads are generally very short, on the range of 50 to 300 base pairs. And sequence reads are also randomly distributed, so you really don't have a read that covers the genome in a really ordered fashion. They're just you just get these random reads, and you have to somehow put those together again. And the sequence quality is relatively low, so there's a high error rate. So you actually have to sequence over and over again the same region to really reliably detect what the sequence is at any given site. And so, for example, you need to have about 50x coverage, 50-fold coverage of any one site in the genome, 50 sequence reads from any one site to really detect a mutation, for example, and distinguish that from some error in the sequence itself, from the sequencing process. And then it can take days to weeks per instrument run. So let's talk now about how massively parallel DNA sequencing works. So the first step in doing massively parallel DNA sequencing is you need to take your DNA fragments that you want to sequence. Those are shown by the DNA fragments up in the upper left. And you have to ligate onto the ends, or covalently attach to the ends of these, what are known as DNA adapters. These are just short double-stranded oligonucleotide sequences. And they come in two different varieties. But these are things you synthesize, and you basically add these to your DNA fragments. And so basically you ligate these onto the ends of your DNA fragments. And what you really want to have here is you want to have your DNA fragments so that each side you have two different types of the DNA adapters ligated onto them. Now you don't have to do anything to enrich for these, but basically these are the only types that are going to be productive in your DNA sequences. So if you have other types forming, it's not a concern. They won't interfere with the sequencing process. And then the next step is you basically take these DNA fragments and you basically heat them up to convert them into single-stranded forms, and then you flow them onto a flow cell. So here's a flow cell, and basically the flow cell is just this substrate that basically has a bunch of these primers. So these primers are just single-stranded forms of these oligonucleotides that are complementary to the DNA adapters. And again, there's two different varieties. One base pairs to one type of the adapter, and the other base pairs to the other type of adapter. And then you flow your single-stranded DNA that you want to sequence onto this flow cell. And what's going to happen is that your DNA fragments are just going to base pair to the sequences that are complementary to the adapters you logated onto the end. And they're just going to drop down and base pair in random locations on the flow cell. So they're going to be spread all over the flow cell, but just in random locations. But the important point here is that it starts off with a single molecule of the DNA that you want to sequence, just base pairing in one spot on the flow cell, and each different spot on the flow cell where something base pairs will be a different DNA sequence. And then what you do is you just flow onto the flow cell all the building blocks for synthesizing DNA. So that means that the four bases, A, C, G, and T, and you also are flowing onto the flow cell a DNA polymerase. And then what happens at this next step is the DNA polymerase is just going to extend the chain in a 5' to 3' direction, which is the direction of DNA synthesis, and you're going to make a duplicate copy of this DNA strand that just hybridized to the primer on the flow cell. And the difference between this sequence that you're just generating and the one that just flowed on the flow cell is the sequence that you're just about to generate is now actually covalently attached to the flow cell. The one that flowed on is just base paired onto there. So if you heat up the flow cell, that one will flow away, as you'll see in the next step. So basically, you're making a complementary sequence. And then after you make the complementary sequence, what you're going to end up with is two sequences that are anchored to the flow cell, because again, the fragment that flowed onto the flow cell base paired to the primer that was basically attached to the flow cell. And so now if you heat the sample up, the fragment that flowed onto the flow cell is just going to come unpaired from its complementary sequence, and it's going to flow off the flow cell. But the other one's going to stay attached to the flow cell because it's anchored there. So these initial steps in the sequencing reaction are actually just amplifying, because you're starting off with one single molecule. You need to amplify the number of molecules that are present on the flow cell at this particular spot to make many, many copies, or else you won't be able to detect the signal when you start doing the sequencing reactions, which we'll come to in a little bit. So you heat up the flow cell, you flow everything off there, and you just have these anchored molecules. And then what's going to happen is the ends of these molecules are going to be complementary to the primers that are on there, and they'll bend over and they'll base pair with the primer that's on there, and then you'll just repeat this process where you'll make a second strand of DNA. So they base pair with their complementary sequences, then you add the building blocks for DNA synthesis again, the four nucleotides and DNA polymerase, and you'll make a complementary sequence to that, using that primer as the starting point for DNA synthesis. And then you just keep repeating this process. You'll flow some nucleotides onto the flow cell again. These will bend over. You'll make new strands. You flow on the nucleotides again and the DNA polymerase. And basically what you end up with is these clusters where basically in a small region of the flow cell, you'll just have amplified the sequences that first drop down there. You'll have many, many copies of that same sequence on that part of the flow cell. So in this case, you have two different clusters, and basically this is the starting point for actually the sequencing reaction. So you have this green cluster and this blue cluster. And again, this is just a small spot on the flow cell where a DNA fragment randomly landed on the flow cell, and you just amplified and made many copies of that same sequence at that one spot on the flow cell. All right, and then from this point, you actually start to do the DNA sequencing reactions, and this is what you're going to actually starting to collect information about the sequence of DNA on the flow cell at that spot. So again, you've got all your clusters in parallel, and now you're going to go through and do some sequencing reactions. You're going to add a primer. It's going to base pair to the primer that's anchored at the flow cell, and then you're going to add the building blocks for DNA synthesis. But they're special building blocks in this case. They're basically nucleotide analogs that are fluorescently labeled. So you have all four of the nucleotide analogs, but they can be distinguished from one another because they've got some sort of label that gives them a different color. So for example, if you, in this first cluster to the left, basically the first base that you would add would be a T, because T pairs with A. And then with the cluster on the right, the first base that you would add would be a G, because G pairs with C. And basically you just repeat this process. This is the 5' end, this is the 3' end. This other strand goes in an anti-parallel fashion. And then basically what you do is after you've added the first nucleotide on there, you capture an image and then you add the next nucleotide. Okay, so the first image, you'd be able to distinguish what base was added because you can distinguish the different bases based on their color. So the first base on the cluster at the left would be a T because that's green, and the first base added to the cluster on the right would be a G because G is blue. So one question that might arise at this point is why is only one nucleotide at a time added to 3' end? And that has to do with the fact that these nucleotides that are used in this synthesis, this massively parallel synthesis, they're modified nucleotides so that you can only add one at a time. So let me just describe what normally happens during DNA synthesis, and I'll describe how they're modified to only allow one base addition at a time. So basically, this is a growing strand of DNA going in a 5' to 3' direction. And this is the 3' end of DNA. This is where the new base is typically added during DNA synthesis. And it's directly, the next base will be covalently linked to this 3' OH that I've highlighted here. And the OH group is absolutely essential for elongation. So let's just imagine the next base to come in here is going to be a G. I'm not showing you the strand that it's pairing with, so just have to take my word for it. The next base pair that comes in is going to be a G. So here's this fluorescently labeled G that would normally come in here and be added by DNA polymerase. But one of the things that distinguishes these nucleotides that are used during massively parallel sequencing is that OH group has a blocking modification on it. So it has an OH there, but there's a chemical addition to that OH which prevents another nucleotide from adding to that position. So basically what's going to happen is that 3' end is going to covalently link itself to the phosphate there, and you're going to add this G to the growing strand of DNA, but you won't be able to add another base to it because that 3' OH of the G that you just added is blocked. It's got a chemical group that's blocking it and preventing another base from being added to it. So then what you do is you've added this first base, you've added a G, and then you basically use a camera to take a photograph of the flow cell after you've added the first base. And then you basically just flush off the nucleotides that float onto the flow cell. You'll add a chemical reagent that removes the blocking group. And then when that's removed, then basically you add the nucleotides again, and the next base gets added on there. And then again, you take a photograph of the flow cell and see what base was added next. And then you just keep repeating this process. So you just repeat these processes stepwise in a stepwise fashion. So when you do this, this is basically what the flow cell looks like. So here's a small portion of the flow cell shown below, and I'm just going to highlight two sequences that are circled. So, and again, I've got the color code for the different nucleotides that you add on each step during the synthesis reactions. And so what you can see here is here at the first step, the first base that was added was a T in both of the circled spots. So we'll look at the first one at the top first. We'll follow what happens with that one. So basically, you remove the blocking group, you add the next set of nucleotides in DNA polymerase. DNA polymerase adds the next base, and the next base it's going to add is going to be complementary to the strand that you're actually using as your template. And so let's say at the next step, the base that gets added, you take a photograph, and what you see is it's a G because you see a blue signal there. And then you just keep repeating this process in a stepwise fashion until we'll just go out 9 steps of the sequencing reactions. And you can just read off that sequence of colors at each step to get the DNA sequence of the strand that you're synthesizing. Okay, so basically at the first position, the first base that was added was a T. The second position where you added was a G. At position 3, that's a C because C is red. At position 4, you added a green one, that's T. And basically, in that fashion, you just read off the sequence. So this gives you the sequence of bases that you add. And again, you do this in a sequential fashion. You add one base at a time. And you can do the same thing for the other circled molecule. But you can see there's a long run of T's followed by a G and then another T. So the sequence of the other one that I've circled is just this long run of T's followed by a G and then a T again. So this is basically how sequencing, or at least next generation sequencing, works. And again, I mentioned right at the very beginning of this slide that this is just one small part of the flow cell. And in fact, this is what a flow cell looks like. And even this is just a very small part of the flow cell. And this is why this is called massively parallel sequencing, because you're basically sequencing many, many, many different DNA molecules all at the same time. You're collecting information on all of them simultaneously. So this is what makes this method so powerful. You can collect lots and lots of information over a very short period of time using these flow cells. So if you want to see a live movie of how the sequencing reactions work, the company that basically makes the reagents that allow for this type of sequencing that I've been describing is Illumina. You can go to a website, And basically, if you just scroll down, you'll see a link to a video that says, how does Illumina NGS sequencing, NGS means next generation sequencing, work? And it'll run through a video and it'll show you basically what I just showed you in the preceding slides, but it'll actually be a movie that's showing you how this all works. Okay, so as I mentioned, massively parallel DNA sequencing is very powerful. You can get hundreds of millions of sequence reads. It's much, much cheaper than the original method, Sanger sequencing, that was used before this. But as I also mentioned, it has some limitations. The sequence reads are fairly short, on the average of 50 to 300 base pairs. The sequence reads are also randomly distributed. I'll mention that on the next slide in more detail, so I'll just hold off trying to talk more about that right here. And the sequence quality tends to be lower than Sanger sequencing, so you need to basically resequence the same site about 50 times each time to get reliable calls of what the actual sequence is at that site. And then, of course, the instruments take weeks, perhaps as much as weeks to run. So I'm going to talk about two of these problems in more detail to sort of illustrate the issue, and I'll also talk a little bit about how these problems are dealt with. So we'll start off with the sequence reads are randomly distributed as the first issue. Okay, so one of the major challenges with next generation sequencing is trying to assemble the individual sequencing reads. And I'll try and illustrate that concept on this slide and the next slide as well. So let's say you have a genomic DNA sample. Perhaps you've isolated this genomic DNA from a tissue sample of some sort. But important point right from the start is you have many, many, many copies of this DNA molecule in your starting sample. And then the first step before you actually sequence is you basically fragment this DNA into smaller pieces. So you're not using a rescission enzyme to do this, you're just randomly fragmenting the DNA. And so you're going to have many, many copies of the DNA now present in the sample. But you've immediately lost the linear order of the DNA sequence in these individual fragments because we don't know which fragment was connected to which fragment, right? So you've lost all that information right from the start. And then what you do is you take these individual fragments and you subject them to next generation sequencing using the methods that I described in preceding slides. And let's say you generate the individual reads that are shown here at the bottom right. So as usual, my example is going to be an oversimplification of the real situation. As I mentioned already, the sequence reads are typically more in the order of 50 to 300 base pairs in length. All my examples, the number of sequences that I'm detecting in each read are all in the order of 10 bases, but I'm just doing that to keep things simple to illustrate my points. So right away, you've got these fragments, but you don't really know how these fragments were connected to one another. But again, the important point here is when you randomly fragment your DNA, you started with many, many, many copies of that same DNA molecule. And so the fragments you generate are going to overlap with one another. And that's the key to reassembling the actual original sequence, that you're going to have a computer that's going to look at each of these individual sequence reads, and it's going to look for overlaps between them. So we'll talk about that on the next slide, how you reassemble these reads. Okay, so let's say you do next generation sequencing, and these are the 7 reads that you obtain from that sequencing process. So again, I'm just going to do this by hand, but in a typical sequencing reaction, you'd have on the order of millions of these reads, and the reads would be longer, more on the order of 50 to 300 base pairs in length. But what the computer does, and this is what I'm trying to illustrate on this slide, is the computer is just going to look at these sequences and look for places where they overlap with one another, and then it's going to align the sequences along according to where they align with one another, where they overlap. So with the example here, you can see that these individual pieces are aligned in a fashion where sequences in common are basically lined up with one another. So you get these overlaps. And then by doing this, the computer can then reassemble the original DNA molecule. So it would basically take from this information that the original DNA molecule from which these various fragments were obtained had the sequence shown at the very bottom. And this bottom sequence is known as a contig. It's just a contiguous stretch of sequence created from the partial sequences. And again, I'm doing this by hand, but this would be typically done by a computer because this is way too much information for a human to process. Okay, on the next slide, I'll illustrate this point a little bit more clearly. So what I'm showing in this slide is each of these green and blue bars represents an independent sequence read from a massively parallel sequencing experiment. And so the bars on average are probably in the order of 50 to 300 base pairs in length. And again, I'm not showing you any sequences. The bars each represent an individual read of that length. And the individual reads are then aligned to one another. This is all but done by a computer. And so they're situated on the slide in a fashion that indicates how they align to one another, the individual reads. And to facilitate reassembly and ensure accurate sequence fidelity, each nucleotide needs to be independently sequenced on average about 50 times. This is also known as 50x read depth. So in other words, you have to sort of sequence any given site in the genome on average about 50 times to really be able to accurately reassemble the sequences and to actually give accurate information as to what the base is at that position. Because again, one thing I mentioned early on is next generation sequencing. It's very powerful. You get lots of information, but it's somewhat error prone as well. So you would need to sequence that same base multiple times because if you only sequenced it a few times, there might be an error there and you wouldn't know what the actual correct sequence is. In general, it does give you the correct sequence, but you have to see it multiple times to be sure what the actual base is at that site. So although there are challenges with reassembling the sequences, it turns out sequencing read depth can also be extremely informative. It can tell you something about the nature of a mutation in a family that you're studying, and that's illustrated on the next slide. So this is actually just showing the sequencing read depth of a particular portion of chromosome 11 from a normal individual. And I'm going to show you two examples of sequencing read depth in subjects who have hemoglobinopathies. Okay, so the one above is the same portion of chromosome 11 showing sequence read depth, and what you'll see is highlighted in blue. This is a portion where the sequence read depth all of a sudden drops off for a little portion along the length of chromosome 11. And if you look at the example on the bottom, you'll see that there's a portion of chromosome 11 where the sequence read depth actually is a little bit higher for over a certain portion of chromosome 11. So what's the interpretation here? Well, it turns out the interpretation here is the one on the top is an example of what you would see if you have a heterozygous deletion, where one of the two homologs of chromosome 11, this portion of the sequence is deleted. That portion has the lower read depth. So it has a lower read depth because you only have reads corresponding one of the two homologs. And the one on the bottom is a case where you have a duplication of one portion of chromosome 11. So instead of having two copies of each sequence all along the length of the chromosome, there's three there. So the odds of actually sequencing that portion of the genome are a little bit higher. So that's why the sequencing read depth is higher in that region. So again, to illustrate this with more pictorially, the one on the top is represented by a deletion, where you have one fewer copy of that portion of chromosome 11, and so the read depth is a little bit lower. And the one on the bottom is a duplication, where one portion of the chromosome is present at an extra dose, and so that portion of the chromosome, you're getting a higher sequencing read depth on that portion. So you can look at sequence read depth and actually learn something about whether there's a deletion or duplication in a particular region of the genome just based on the read depth part of it. Okay, so going back to this cost per genome curve graph that I showed you earlier, one of the things it turns out is as of 2018, it costs about 1 cent per million bases to use this sequencing approach. So it's really super cheap, and the cost is even lower now. It's 2020 now. It turns out the human genome can be sequenced for about $3,000 at this cost, and so the idea has sort of arisen whether you can actually identify disease-causing mutations simply by sequencing the entire genome. You can skip all this linkage mapping approach and just not even try to bother to locate the gene into a very small portion of the genome. Instead, just bypass all of that and sequence the entire genome and look for the disease-causing mutations. So again, can we just sequence affected and uninfected individuals to look for the disease-causing mutations? Sequencing has gotten so cheap that this should be a pretty straightforward process, right? So the basic idea is this. Let's say you've got some sort of heritable disease. Individuals with the disease are highlighted in the black shading. Normal individuals in this family are in white. And basically what the idea is, you can just sequence disease individuals and then also sequence DNA from normal individuals. And then you just kind of compare the two sequences and you look for the disease-causing mutation. Can you do this? Well, the answer is yes, you can do it. I'm going to show you an example of where it was done. But there are some challenges that need to be addressed if you're going to use this approach. So let's go back to a slide where I told you something a long time back. So one of the things I told you in an early lecture is that it turns out that there's a lot of polymorphisms in the human genome. These are the things that are used for doing linkage analysis. So I gave you an example of the UW student population. Just to describe what a polymorphism is, it's just a place in the genome where you see variation in different individuals in the population. And I think I briefly alluded to this, but I'll sort of make sure that I'm clear on it right here. If you compared homologous chromosomes from any two individuals, or in fact, even if you compared any two of your homologous chromosomes, in other words, if you compared your chromosome 2 that you inherited from your mother to the chromosome 2 that you inherited from your father, and you compared the sequences to one another, what you'd find is, on average, about once every 1,000 base pairs, you'd see a difference in the sequence between those two homologous chromosomes. So on average, between any two homologous chromosomes over the entire genome, you see a base change once every 1,000 base pairs. So let's do a little bit of math here. So if there's one polymorphism on average per 1,000 base pairs, and the human genome is 3 billion base pairs in length, if you compared all the homologous chromosomes to one another, all the homologous chromosomes you inherited from your mom and from your dad to one another, you would detect about 3 million differences between them all told. So that's a huge number of differences. So if we use this approach that we talked about, basically comparing the sequences of two different individuals, you detect about 3 million differences between them. So the question then arises, which one is the disease-causing mutation, right? Only one of them is presumably the disease-causing mutation, but you're going to have to figure out which one that is amongst these 3 million different base changes in these individuals. But one way we can make this a little bit easier is if we don't actually compare the entire genome between these two individuals. What if we just compare the portion of the genome most likely to bear the mutation of interest? Is there a way we can do that? So yes, there is a way we can do that. It turns out that only about 2% of the human genome codes for protein, as I talked about earlier in this lecture. So only 2% of the human genome is devoted to exons. And it turns out most human disease mutations affect just coding exons or splice sites, the parts of introns that reside right next to where the coding exon is. So, but what if we can just sequence the exons? What if we just sequence that part of the genome? This is also known as the exome, the expressed portion of the genome. So if we just sequence the exons, then we would only be sequencing 2%, and that would greatly reduce the magnitude of this difficulty of distinguishing the mutation from naturally occurring polymorphisms between any two homologous chromosomes. So is there a way we can do that? The answer is yes, there is a way to do that, and it's called exome sequencing. The way this works in simple straightforward explanation is you just synthesize a suite of oligos that happen to match all the coding exons, all right? And these suite of oligos that match the coding exons are also known as probes or capture oligos. And you synthesize this capture oligos with some sort of bead attached to them that allows you to actually retain them and wash away the sequences that you don't want. So let's say these capture oligos have magnetic beads on them, for example. So what you do is you take your fragmented source DNA, you denature it to convert it to single-stranded form, and these capture oligos are already in single-stranded form, and then you hybridize them together, you mix them together, and the capture oligos are going to base pair with the sequences from the fragmented source DNA that they're complementary to. And again, if your capture oligos just correspond to the exons, they're only going to base pair with exonic DNA sequences. So if you mix them together, again, the capture oligos are going to base pair with just the exon sequences. And then if you suspend all of this, as this is all done in solution, you suspend this in a magnetic field, the beads are going to be held back, and the fragments of DNA that are not base paired to these capture oligos, they're going to wash through the column. So you discard all the fragments that don't base pair to your capture oligos, and you retain the capture oligos with the DNA sequences that they're complementary to, which in this case would be just the exons, the coding exons. So you discard all the fragments that don't base pair, and you capture all the coding exons, and you subject them to sequencing analysis. And then you're going to look at those sequences, compare them to control individuals, and you're going to look for mutations that are responsible for the disease that you're studying. On this and the next few slides, I'll talk about an example of using exome sequencing to identify the gene responsible for Miller syndrome. So just a little bit about Miller syndrome. This also has a technical name. It's a little bit hard for me to pronounce. Post-axial acrofacial dysostosis. It's a rare autosomal recessive multiple malformation syndrome. So a couple of examples of what this syndrome phenotype is like. So it's associated with facial malformations, chest malformations, there are upper limb defects, as well as lower limb defects. And basically, to identify the gene defect responsible for this syndrome, the group that did this work sequenced exomes from 3 different families that were segregating Miller syndrome. And this work was actually done here at the University of Washington. The individuals that did this work is Sarah Nig in the laboratory of Jay Shinduri. I may not be pronouncing her name properly. And this group is actually here in our home department, Department of Genome Sciences at University of Washington. So let's talk a little bit about what they did. On the next slide, I'll summarize the results of their analysis. Okay, so this slide summarizes the results of the analysis that were carried out in the Shinduri laboratory. So what they did here is they basically just, after they did the exome sequencing, they basically asked how many genes in the genome had some sort of mutation that gave rise to an amino acid alteration relative to the wild type. And they were looking for amino acid alterations in both alleles of the gene because, again, the Miller syndrome is a recessive disorder, so you'd expect both copies of the gene to be defective, to have a mutation. And so when they did this, they calculated the total number in family 1, the total number were common between family 1 and family 2, and the total number that were in common between all 3 families. So again, the names family 1, family 2, and family 3 are just arbitrary, but I'm just going to use these terms anyway. So they found in family 1 there were 2,278 genes in the genome that had an amino acid alteration in both copies. And then of these 2,278, 1,740 of them were in common between families 1 and 2. And then the number that were in common between all 3 of the Miller syndrome families were only 1,461. So the number has been reduced by looking at how many were in common between the 3 families. But 1,461 is still a relatively large number of candidate genes to be working with. So they had to do some additional analyses to reduce this number even further. So the next thing they asked is, of these gene alterations in the Miller syndrome families, how many of these gene alterations are not among known common SNPs? I've already told you that there are lots of naturally occurring polymorphisms in the population. These are just changes in the DNA sequence between different individuals in the population. Most of these have no phenotypic effect. And so basically they asked, of these amino acid alterations detected in the Miller syndrome families, how many were not among known common SNPs that are just in the wild type population? And that greatly reduces the number. So only 40 that were detected in the Miller syndrome families were not detected even just in the normal population. And then of these 40, only 16 were in common between families 1 and 2, and only 12 in common between all 3 families. And then they did one further experiment to kind of further try and reduce the numbers. They basically sequenced 8 control exomes. These are from normal individuals. And they asked, of the amino acid alterations they detected in the Miller syndrome families, how many were also not present in these 8 control exomes? And that again also reduces the number, right? So they end up finding only 47 genes that were specific to family 1, these gene alterations that were specific to family 1. And then again, when they sort of asked how many of those are in common between family 1 and 2 and all 3 families, that really also greatly reduces the number. And finally, they asked how many of the gene alterations that result in an amino acid change in the Miller syndrome families were not among common SNPs or in these 8 control exomes. And this dramatically reduces the number. So it's only 8 in family 1, and then it turns out when you ask of those 8 how many are in common between family 1 and 2 or in all 3 families, it narrows down to 1. So this last gene that you're left with is presumably the Miller syndrome gene. So what does this gene encode? So this is the Miller syndrome gene. It turns out the gene is called DHODH. It encodes an enzyme called dihydroorotate dehydrogenase. And this is just an enzyme that's responsible for pyrimidine biosynthesis. And I'm showing you the structure of the gene down below. And it has 9 exons. The coding exons are designated in blue. And then all the mutations that were detected from exome sequencing are illustrated with these red asterisks. So there were a number of mutations that were detected, right? Because they sequenced the exomes they actually sequenced the exomes for 4 different individuals from 3 different families, it turns out in the end. So an interesting side point is that it turns out mutations have a DHODH gene as well, and mutations in that gene in Drosophila give rise to wing and leg malformations. So it looks like Drosophila might actually be a pretty good model system to study the mechanisms underlying this disease and maybe to try and develop treatments as well. So the key features were this. There were multiple independent families segregating Miller syndrome that were available. So that allows you to basically look for things that are in common between different families. It turns out Miller syndrome is an extremely rare disease, and so you're unlikely to have asymptomatic carriers in the control population. That's also very important. Miller syndrome is also recessive. It's relatively rare for any one gene to have two candidate gene defects. You have to have one in each allele. That's not also detected in the control population. So that helps reduce the number of candidate genes down quite substantially. And another feature that I haven't really talked about in any detail is that the individuals that were used in this study were of European ancestry, and it turns out there are just fewer rare variants in Europeans than in other populations, such as East Asians and Africans. So this helped with the study a little bit as well. So just to summarize what I've told you, it turns out linkage mapping can identify the approximate position of a gene conferring a disease or a trait, and that facilitates the discovery of the gene itself because you know that you only have to look in a small region of the genome to identify that disease-causing mutation. But you can also apply whole exome or whole genome sequencing, and you can bypass linkage analysis altogether and directly identify causative mutations for Mendelian disorders. It's thought that as few as 3 unrelated patients are sufficient if you have a recessive disorder. The cost is dramatically lower, and it even works well for very rare disorders. It's not going to work in all cases because not all diseases are recessive. You're not always going to have 3 unrelated patients. So, but in many cases, this approach works really very well, and it's much cheaper and much faster. So why is identifying disease-causing gene mutations useful? So there's a number of reasons it's useful. It can help in diagnosis. So for example, if you can actually sometimes sequence to see if you're likely to develop a disease at some point in your life. Sometimes knowing what the allele is can tell you how severe the disease might be. So there's a number of reasons why it can help you in diagnosis. It may also help to reveal mechanism of disease by sometimes if you know the function of the gene, it can tell you something about how to treat the disease. It can also lead to preventative strategies. One of the best examples of that is phenylketonuria. This is a disease where a mutation in a particular gene that breaks down phenylalanine, an amino acid, is defective. And because that gene is defective, you can't break down phenylalanine. It builds up to very high levels from the diet, and that could lead to mental retardation. So there's a very simple treatment for this, which is that when a baby is born that has this disease, you basically just leave phenylalanine out of the diet as much as possible, and people who are treated that way can actually live a completely normal lifespan and with no mental retardation whatsoever. But it turns out many of the gene defects that you detect don't have obvious treatments, and in such cases the ideal solution is to basically just replace the defective gene or to do something to kind of correct the nature of that defect. And we'll talk about 2 different approaches for doing that people are trying out in the next few slides. Okay, so we'll talk about 2 different approaches for correcting gene defects that cause disease. One approach we'll talk about first is gene therapy. And the example we'll talk about here is this individual pictured in this photograph. It's a young boy who has this disease called severe combined immunodeficiency disease, or SCID. It's also known as the bubble boy disease, based on the fact that there was another individual who basically lived his entire life in a bubble or in a protective bubble because he just didn't have any ability to fight off pathogens. This is actually a collection of genetic diseases associated with defective immune cell development, and you have really severe enhanced vulnerability to infection. So there are a number of gene mutations that can give rise to this disease. And then here's the individual who this disease, the sort of common name bubble boy, was named after, David Vetter. Turns out David Vetter had a sibling who died at 7 months from this disease. He also inherited this disease, but he was placed inside of a protective plastic bubble right at birth, and he lived his entire life in that plastic bubble. He died after developing a lymphoma following a bone marrow transplant from his sister. It turns out his sister had a Epstein-Barr virus infection, which was really non-pathogenic in normal individuals, but it ended up being fatal for him. So, okay. So let's talk about how the gene therapy was used to correct SCID in this young individual that was pictured in the previous slide. So basically what people did is they took bone marrow cells from the SCID patient that had an IL-2R mutation. As I mentioned on the preceding slide, there are mutations in many different genes that can give rise to the SCID syndrome. IL-2R is one of these genes. So they took bone marrow cells from this individual, and then they also at the same time they engineered a modified form of the HIV virus. And basically what they did is they replaced a portion of the HIV genome with a normal copy of the IL-2R gene. So it's an HIV virus that carries the IL-2R gene. Okay, so it turns out the reason they used the HIV virus in this case because HIV expresses on its surface a protein that binds to a receptor that's expressed in some types of immune cells, CD4 type cells. And it turns out if the receptor is a protein, it's a complex of 3 different proteins, basically, one of which I'll come back to a little bit later in the lecture, CCR5. I'll have a little bit more to say about that later. But basically, the reason they use HIV was because it is very efficiently taken up by some immune cells, because the immune cells have a receptor that binds to a protein on the surface of HIV, and that's how it imports this virus inside the cell. So what they did is they used the modified virus to infect bone marrow cells, and they grew these cells in culture for a short while, and then they returned the cells to the patient. And what they found remarkably is basically the idea here is that these cells are infected by this virus that has a normal copy of the IL-2R gene, and basically what it did is it restored immune function to 7 out of the 8 individuals that received this treatment. So this is a very promising treatment for treating this disorder. But it's important to sort of point out that this approach has been tried in the past, and sometimes it's had promising initial findings, but the findings start to wear off. But apparently, this young boy that was pictured in the preceding slide, he's had the treatment for over a year, and he's still actually doing quite well. So it has a great deal of promise and a lot of potential. So the other approach I'm going to talk about that you probably heard a lot about from the news is CRISPR-Cas9 gene editing. So this is actually a really flexible gene editing system that was actually discovered in studies of bacterial immunity. So a little bit about the nomenclature. The term CRISPR stands for clustered regularly interspaced short palindromic repeats. And it's because if you look at a portion of the genome of bacteria that contain CRISPR, the portion of the genome basically has clustered regularly interspaced short palindromic repeats. So these yellow boxes are, and this is just supposed to represent a small portion of the bacterial genome, I should mention that. The yellow boxes represent the short palindromic repeats. So if you look at the sequences of palindromic repeats, what they look like is something like this, where the palindromes are sequences that read the same way both in the forward and the reverse direction. So for example, if you read the top strand in a 5' to 3' direction, you see it goes G, C, A, C, C. And if you read the bottom strand in a 5' to 3' direction, it also reads the same way, G, C, A, C, C, right? So these are known as palindromic sequences because they read the same way 5' to 3' on the top strand as they do on the bottom strand. So it has a number of these palindromic repeats. They're the same sequence repeated, but they're also interspersed by these unique sequences. And then as far as the Cas9 component to the CRISPR-Cas9 gene editing system, Cas9 is a separate gene somewhere located elsewhere on the chromosome, and it encodes a protein, which is a nuclease, it turns out. And nucleases are proteins that cut DNA in specific locations. So these are the 2 components of this CRISPR-Cas9 system. So how does the system work? Well, it turns out an important clue came when people started looking closely at the sequences, these unique sequences. It turns out the unique interspaced sequences match precisely to sequences in phage DNA. Phage are like bacterial viruses. So again, the clue was the interspaced unique sequences matched very precisely to phage DNA. And the different colors represent sequences that are present in different phage particles, different types of phage that infect bacterial cells. So let's talk a little bit about how the system works. And this is actually the model slides that I'm going to be showing you is basically based on work that was done by researchers over a number of years, basically. So what happens normally if you have a phage infection in a bacteria that doesn't have this CRISPR-Cas9 system, the phage actually injects its DNA inside the host, And then basically it usurps all the cellular machinery to start making phage products, basically. It transcribes and translates the phage genes, makes phage proteins, and then you start to assemble new phage particles inside the bacteria. And then ultimately, this leads to the lysis of the bacteria and the release of new phage particles to the environment so they can infect more bacteria. Okay, so that's what happens if the bacteria does not have the CRISPR-Cas9 system. What happens when the bacteria had the CRISPR-Cas9 system? So in the presence of CRISPR-Cas9, the situation is a little bit different. The phage injects its DNA just like it did in the previous slide. But if this bacteria has the CRISPR system, basically the CRISPR RNAs are being made all the time, and the Cas9 protein is being made all the time. And what Cas9 protein does, it assembles with the CRISPR RNA. So I'm just going to show you assembling with one of the CRISPR RNAs here, the one that's the same color as the phage DNA. But it turns out the Cas9 protein assembles with all the different CRISPR RNAs. Each of these RNAs has a sequence that's the same as the sequence in a different type of phage. But in this case, I'm just showing it assembling with the red one, which happens to be the same sequence in the phage DNA that was just injected into the bacterium. And then what happens is that Cas9 protein in complex with the CRISPR RNA, it goes to the portion of the phage DNA that matches the sequence in the CRISPR RNA, and then the Cas9 protein catalyzes the cleavage event and cuts the phage DNA in two pieces. So that basically inactivates the phage, and it prevents bacterial lysis. As an aside, I have to say something you guys talked about in the first half of the class, the use of restriction enzymes to make libraries and so forth. So it turns out restriction enzymes were also discovered in studies of bacterial immunity, and they act similarly to the CRISPR-Cas9 system. So I'll just describe that briefly before I continue on and talk about how the CRISPR-Cas9 system is being used for gene editing. So in bacteria, many bacterial strains encode a restriction enzyme, and what happens is that the phage comes in and injects its DNA. If the phage DNA happens to have a site that the restriction enzyme recognizes, the restriction enzyme will go to that site and it will cut the DNA, and it will also inactivate the phage particle as well and prevent lysis. So it works in a very similar fashion to the CRISPR-Cas9 system. So it's interesting to note that two of the major ways that people do gene editing, the two of the major pathways that are used to do gene editing were discovered from studies of bacterial immunity. That's a pretty remarkable thing. Okay, so getting back to CRISPR-Cas9 system, let's talk a little bit more about how the system works. So to look at this, we're gonna blow up what this looks like when the Cas9 protein in complex with CRISPR RNA, when it's bound to the phage DNA. What does it look like at the molecular level? So if we blew that up, it would look something like this, where it turns out the CRISPR RNA is basically hybridized to one of the two strands of the phage DNA. So it actually goes, the CRISPR RNA in complex with Cas9 goes to the portion of the phage DNA that it's complementary to. The double strand of the phage DNA opens up, and the CRISPR RNA base pairs with one of the two strands, the strand that it's complementary to. And then basically the Cas9 protein, after this base pairing occurs, the Cas9 protein, which is the nuclease, it cuts the phage DNA on both of its strands in the same location. Okay, so a lot of this work that was done to characterize how the CRISPR-Cas9 system works in bacterial immunity was worked out by these two individuals here, Emmanuelle Charpentier and Jennifer Doudna. And they actually, after they discovered a lot of this stuff, they basically asked two really important questions. So one question they asked was, can we modify CRISPR to recognize any sequence of interest? So in other words, can we change the sequence of the CRISPR RNA to anything that we desire so it would cut any DNA that we wish to have digested? And the other question they asked was, I didn't mention this, but for this system to work, you have to also have this tracer RNA. This is this black RNA that's base paired to the CRISPR RNA. These are two separate RNAs. I didn't describe that, but the tracer RNA is also important for function. It's not really immediately clear what role it plays, but if you don't have it, you don't see this cleavage event occur. So they also asked, can we combine CRISPR and tracer into a single molecule? So they basically just could make one single molecule that has both of these sequences. So they just join these together basically to do that. So the tracer CRISPR hybrid RNA is called a guide RNA or a gRNA. So that's the term I'll be using in the remaining slides. And turns out the answer to both of these questions was yes, that you could do these things and the system worked. So basically, you can design the CRISPR RNA to correspond to any sequence of interest, and in complex with the Cas9 protein, it'll deliver the CRISPR RNA to any sequence of interest, and it'll cleave at those sites. So now on the next slide, I'll describe a couple of approaches people are using to use the CRISPR system. So this CRISPR technology allows genetic alterations basically anywhere in the genome that you wish, in any cell type that you want to perform this sort of gene editing in. And there are two approaches that are typically used for CRISPR-Cas9 gene editing. Both approaches start the same way. Basically, you design a guide RNA that targets the Cas9 nuclease to the specific location you want to create these cuts at in the genome. And so the first step is you create these double-strand breaks. And then one approach is gene silencing. Typically, when you create double-strand breaks in the genome, what oftentimes happens is one of two things. Either you have some of the nucleotides at the broken ends that get chewed away, or you have random insertion of some nucleotides at the two broken ends, and then that's followed by the two ends joining again. And typically when that happens, either removing some of the bases or adding extra bases, I've illustrated it here by adding a few extra bases, that ultimately results in disruption of the gene. So this is an approach that's in common usage if you want to create a model of some sort of disease in some sort of model organism. So for example, if there's a mutation in a particular gene that inactivates the gene in humans and that gives rise to a disease, and you want to create a gene inactivation of the corresponding gene in mice, you would use this gene silencing approach to do that. So that's actually quite common, and it's a common way to create model systems to study disease now. The other approach that's in common usage is gene editing. And so again, the first step is the same. You just create a double-strand break. And one of the ways that the cells can repair that break is if you can supply the cell with some sort of repair template, then what it'll do is it'll use homologous recombination to basically use the repair template to replace the double-strand break portion of the genome. And this is also known as mitotic recombination because typically the types of recombination we've talked about take place in germline, and it gives rise to the formation of gametes. But this is actually something that does occur in mitotic cells as well, but it's only done in a repair pathway. If it's got a normal template that it can use for repair, it'll use that. So the way people are envisioning this being used is that, for example, if you have some sort of human disease and it's caused by some sort of mutation in a gene, you can potentially create a double-strand break in that gene region. And then if you supply a repair template as well, the double-strand break will be repaired using the repair template, and you can essentially replace the defective portion of the gene with whatever sequence you want to put in there, presumably the wild-type portion of the gene. So this is a way you can actually do gene repair. So CRISPR-Cas9 genome editing, to my knowledge, has not yet been used in any human disease context, but there's clinical trials underway in many different contexts. So there's currently, and this slide's a little bit out of date, there's currently clinical trials in progress using CRISPR-Cas9 gene editing to treat some of the following diseases. So I won't read all of these, but there are a number of cases out there. I'm sure that there's additional cases since I prepared this slide about a year ago, so there's probably other more recent cases where people have tried to use this as well. However, one of the concerns with using CRISPR-Cas9 editing involves its targeting specificity. So it seems to be fairly precise in terms of the sequences it's directed to by the guide RNA, but in a lot of experiments people have done, they've found that in fact it actually can sometimes go to a site that doesn't have perfect complementarity to the guide RNA. So there's some question about like how specific it's going to be. Because if you want to do some sort of gene editing event, you want to make sure it's creating the cut where you want it to create the cut. You don't want that guide RNA and Cas9 being delivered to other parts of the genome and creating mutations elsewhere. That could create a lot of problems for you. And on the next slide, I'll describe a use of CRISPR-Cas9 to do gene editing in human embryos that's a very highly controversial issue. You may have heard about this in the news. There's also been a recent controversy involving CRISPR-Cas9 gene editing. It's based on work that was done in China. So this Chinese researcher, I'm not sure how to pronounce his name, He Jiankui, something like that. He basically edited embryos, human embryos, and created the first gene-edited babies using CRISPR-Cas9 gene editing. So this is a little bit controversial for several reasons. First of all, I think a lot of people felt it wasn't quite ready to be applied in humans because we still didn't really have a good sense of how many off-target sites you might hit. So he might make the correction he wanted to make, but he might also at the same time have cleaved the DNA in other places and made new mutations. It's just not clear. So let me tell you what he tried to do. So he actually used CRISPR to generate a null allele of CCR5 in embryos. So we talked about this already. I said I would come back to it a little bit later. So CCR5 encodes a 7-transmembrane domain G protein coupled receptor for cytokines. And it turns out that there's actually there's an allele of this in the population where a portion of the sequence is deleted. So about 10% of the population of European ancestry bears an out-of-frame 32 base pair deletion at this site that's I've highlighted in yellow. So the protein is not, only a small truncated portion of the protein is made, and the protein that's made is not functional. It turns out that homozygotes for this allele are resistant to HIV infection, and the reason why is based on something I told you already. So it turns out, as I mentioned, that the HIV virus, it basically uses this CCR5 co-receptor to gain access to the immune cells. So if you don't make the CCR5 protein, it can actually enter immune cells. So it's basically harmless to people that don't make this receptor. It's really not clear whether people who don't make this receptor have any other associated phenotypes, except the fact that they're immune to HIV infection. So there's CCR. Okay, so why is this work controversial? There's actually several reasons why it was controversial. One of the reasons is that they were editing embryos. So this leads to germline changes that will lead to heritable changes. So for example, the children that receive this editing change, they'll also pass this change on to their offspring as well. So this is sort of a controversial area. Usually it requires a lot of discussion among ethicists, what's permissible, what's not permissible. So that's one of the things that made this sort of controversial. Another thing that made it controversial is something I already alluded to, which is the targeting specificity of CRISPR-Cas9. So we still don't really know how specific it is, and we know for sure that there are some off-target sites, places in the genome that you didn't want CRISPR-Cas9 to cut that sometimes get cut. So that's still a bit uncertain. So I think a lot of people feel it's a little bit too early to be using it to edit embryos before we really understand all the sites that it cuts that are off target and try and improve its fidelity as much as possible. Another thing was that the logic of targeting CCR5 was very much in question because, again, this is an editing change that wasn't going to change the fate of these individuals, embryos. They didn't have some terrible disease that awaited them in the future. So the logic of targeting CCR5, which is basically a targeting event that's not really a necessary thing that needed to be done, that's also very much in question as well. Finally, the specific change that was engineered to inactivate CCR5 was actually different than the one that's found in nature. So that polymorphisms that's found where a deletion of part of the coding sequence is detected in people of European ancestry, that was not the change that was made to inactivate CCR5. It was a much bigger change. The specific nature has not been revealed to my knowledge, but apparently it was a much larger deletion. So it may be that really just inactivating the gene is really the issue here, but it might also be the case that just making a small truncated product like the naturally occurring mutation does versus whatever was engineered into this one could have a different phenotypic effect. So that's also sort of controversial. And that's it for today's lecture.